Hello everyone, my name is Paul Kim. I am CFO of PharmAbcine. I'm here to introduce PharmAbcine to overseas investors. Before I begin, I'd like to express my deep gratitude to IR Council of the Korea Exchange for this presentation opportunity. This presentation will be divided into two parts. In the first part, I'll talk about the company overview, and in the second part, our biospecialist Sung-Jun will talk about the pipeline assets. PharmAbcine is a clinical stage biotech company specializing in antibody therapeutics. We were established in 2008. Our marquee investors included Novartis Venture Fund and OrbiMed. Attracting investment from these big names like Novartis and OrbiMed meant a lot for us because their investment says a lot about our science as well as business prospect. The company went public in 2018 and became a KOSDAQ listed company. Over the years, we have developed a robust R&D platform capable of producing multiple first-in-class assets. Further, having worked with global pharmas and world-class hospitals, we now have accumulated extensive and differentiating experiences in R&D and clinical studies. With well-established R&D platform, global partnership experiences, together with world-class management team, I strongly believe we are ready for big jump. This slide talks about the factors differentiating PharmAbcine from other antibody therapeutics companies. We already mentioned about our early funding from big names like Novartis and OrbiMed and what it means. In addition, I would like to emphasize our current and prior research collaboration with global pharmas, including MSD, Roche, and Sanofi. In fact, we currently have 2 ongoing clinical trials with MSD. As for the R&D platform, we take pride in our proprietary antibody library called HuPhage. It contains more than 100 billion fully human antibodies. Thanks to this amazing diversity, we now have 3 first-in-class molecules in our pipeline. We have a world-class management team with not only extensive drug development, everyone has extensive drug development experience, but also a powerful network of global key opinion leaders. For example, we have Dr. Napoleon Ferrara as a SAB member, Science Advisory Board member, and he's the scientist who developed Avastin. We also have Dr. Bob Kerbel, world-renowned authority in tumor angiogenesis field, helping us as a consultant. We strongly believe that our network with these global key opinion leaders will most likely enhance our ability to develop and license out our pipeline assets. There are 3 different paths to our long-term growth strategy. 1, progression from tumor angiogenesis to vessel normalizer within oncology. 2, expansion of vessel normalizer from oncology to non-oncology. 3, entrance into immuno-oncology. Our journey began with tumor angiogenesis. Olinbecimab, our lead molecule, blocks tumor growth by inhibiting VEGF and VEGFR2 pathways. Tumor angiogenesis has been around for more than 30 years. So it is time for something new and or better. Our primary interest is shifting towards vessel normalizers. Our preclinical molecules like PMC403 and PMC42 normalize pathological vessels that are normally leaky and disorganized. Vessel normalizers reduce tumor growth by improving delivery of anti-tumor drugs and immune cells. The most wonderful thing about vessel normalizer is its expandability. There are so many non-oncology indications for which PMC-403 can be used. Such an expandability, tremendous growth potential for PharmAbcine in the longer term. Finally, we are entering the booming immuno-oncology arena with PMC-309. PMC-309 is a unique first-in-class molecule that targets VISTA. Unlike other immune checkpoint inhibitors, PMC-309 activates T-cell indirectly via MDSCs. PMC-309's unique mode of action allows us to develop both mono and combo therapies for cancer patients. Next two slides will show how well positioned PharmAbcine is for the changing business environment. There are two dominant trends in oncology. First, there is a rapid increase in the number of combo studies involving immuno-oncology drugs, or IO drugs in short. Two, there are immense needs for new IO drugs. What is causing these two trends? It is the low response rates of existing IO drugs that's causing all these mega trends. As shown in this chart, the IO drugs have less than 30% response rates for most oncology indications. This is including the combo studies as well. Because combo therapies can significantly increase the response rates of IO drugs, the number of combo studies has risen drastically in recent years. The most popular combo molecule for IO drugs is tumor angiogenesis inhibitors. This is wonderful news for PharmAbcine because we are the expert in tumor angiogenesis. We have olimbesimab, which has the best-in-class potential, thanks mostly to its safety profile. As for the rising needs for new IO drugs, the company has PMC-309, a VISTA-targeting T-cell activator. In sum, the key message here is that PharmAbcine is well positioned to benefit from these two dominant trends. This slide shows two things. One, a rapid growth in combo studies, and two, the popularity of tumor angiogenesis inhibitors in combo studies with IO drugs. The chart on the left shows a rapid increase in the total number of clinical trials involving IO drugs, like Keytruda. As you can see, the increase predominantly comes from combo studies. For example, the total number of clinical trials involving Keytruda, scholastic name is pembrolizumab, rose by 642 trials. That's an increase of 115% over 2017 and 2020. Of these 642 increases, combo studies accounted for 84%. So substantially, all of the increases came from combo studies. The chart on your right is showing that the most popular combo partner for these IO drugs in 2020 was VEGF or VEGFR targeting molecules, i.e., tumor angiogenesis inhibitors. The popularity of VEGF or VEGFR targeting molecules appears to be rising. In 2019, VEGF or VEGFR targeting molecules were number 3 popular combo partner. This slide shows the expandability of vessel normalizer. Just look at the variety of indications that our PMC-403 can cover. Personally, PMC-403 is like a family panacea. My family members have glaucoma, Alzheimer, lymphedema, and rheumatism. Many research suggests these indications can be treated by a type 2 activating molecules like PMC-403. So what about the drug development? Our drug development program for PMC-403 will start with eye diseases like wet AMD or DR, and then we'll gradually expand to other indications including CKD, chronic kidney disease, and sepsis. PMC-403 is already getting some attention from scientists around the world. June last year, we signed a research collaboration with NIAID, National Institute of Allergy and Infectious Diseases in the US. Under the agreement, NIAID will perform a preclinical study for SCLS, or systemic capillary leak syndrome, with PMC-403. Earlier this year, we also signed a research collaboration with LegoChem Bio, a leader in antibody drug conjugate, ADC. LegoChem will perform a preclinical research to find out if PMC-403 can improve delivery of their ADC to tumor. It is highly encouraging for us because PMC-403 is getting already noticed by these drug development experts around the world. And this PMC-403, we expect this will provide tremendous growth potential in the longer term. Last year, we set up a 100% owned subsidiary in the US, and we named it Wincal Biopharm. We find this to be one of the most important moves for our long-term strategy. Why is it important? It is important because it can unlock or expand our existing R&D platform to non-oncology, and further, it will also unlock the full potential value of our existing pipeline assets that could be used for both oncology and non-oncology indications. PharmAbcine has long focused on developing oncology drugs. Therefore, our knowledge and resources in developing non-oncology drugs are quite limited. Further, it is extremely difficult to find good R&D professionals in Korea at the moment because there has been a long-lasting boom in the bio industry, and this has been going on for many, many years. And we don't expect this trend to go away anytime soon. So to overcome our weaknesses and difficulties, we established Wincal. We were very fortunate to staff Wincal with highly experienced vascular disease experts in the US. In fact, we hired a team of vascular disease experts from Amgen. Wincal will focus, going forward, on developing drugs for non-oncology indications, especially vascular diseases. It is also developing eye drop formulation. If successful, this is going to be groundbreaking for us because we are also developing PMC-403 for eye diseases as well. Well, this just about sums up our company overview. And I would like to invite Seongjun to the podium, and he'll discuss the pipeline assets. Thank you.
Hello everyone, my name is Seongjun, and I'll be covering 5 things to know about our R&D pipeline. There are 3 ongoing clinical trials, 2 combo studies, and 1 mono study, all of which involve olinvestmab. There are 2 phase 1b combo studies in pembrolizumab for MTMBC and RGBM patients. Of the 2 combo studies, the MTMBC study will proceed to the next phase in 2021. The RGBM study will not be proceeded to the next phase for now. We recently submitted IND application for IIT involving olinvestmab and capecitabine, which will begin soon. The PMC-40309, PMC-403, and PMC-402, all of which are our first-in-class molecules, will enter the clinical stage over 2021 and 2022. Here's our pipeline overview. First, we have 2 combination trials ongoing in Australia. Then we have olinvestmab mono for Avastin refractory RGBM patients currently ongoing in the US and Australia. We are soon going to have an IIT study ongoing in combination with olinvestmab and capecitabine for MCRC patients. Capecitabine is a chemo drug used to treat many oncology indications. Then our other preclinical assets, such as PMC09, PMC403, and PMC402, will undergo a global phase 1 trial over the next year. Next, I'll be presenting the 5 things to know about our olinvestmab. Olinvestmab is the lead molecule with best-in-class potential. It is a tumor angiogenesis inhibitor, and it blocks VEGF and VEGFR2 pathways. Olinvestmab is able to block all VEGF signals, including VEGF-A, VEGF-C, and VEGF-D, unlike Avastin. It does so by targeting the receptors or VEGFR2 instead of ligands. In the clinical trial, olinvestmab has shown superior safety profile compared to other competing molecules such as Avastin and Cyramza. We were able to gather encouraging interim results from olinvestmab pembrolizumab combo trials for mTNBC. Here's the competitive landscape of olinvestmab. So far, we were able to discover that olinvestmab has a different mode of action from Avastin, but very similar mode of action with the Cyramza. Olinvestmab targets VEGF-A, C, and D by inhibiting the VEGFR2 receptors, while Avastin can only block VEGF-A. Olinvestmab has superiority in the side effects when compared to Avastin and Cyramza. Olinvestmab has only showed less serious adverse events compared to those two competitors. Here is the clinical development of olinvestmab. First, we have a phase 2 Avastin non-responding RGBM study ongoing, which started in November 2019. We were able to gather 17 patients out of 36 as of now. Then we have an ongoing trial of phase 1B study for RGBM in combination of olinvestmab and pembrolizumab, which started in January 2019. Then there's another olinvestmab and pembro combo study ongoing for mTNBC patients. It began in December 2018. Here is the interim results that we presented at the SABCS. In terms of safety, all adverse events were manageable in the hospital settings. No DLTs were found in all the patients. There were some treatment emergence and some serious events, but they were well tolerated. In terms of efficacy, in the high-dose cohort, we were able to find 50% ORR and 67 DCR. In the high-dose cohort, one of the patients with PR had a CR in the target lesion. Here's the detailed graph of our interim result. The left graph is for the 12 milligrams per kilo of olinvestmab. We should be focusing on the right graph, which is the 60 milligrams per kg. This cohort has shown 50% PR. If you look at the green graph, the patients showed the CR in the target lesion. However, this patient was labeled as PR because another tumor was detected in a non-target lesion. Based on this encouraging result, we were able to gather rational data enough to proceed to phase 2. Next, I would like to move on to PMC309. PMC309 is our first immuno-oncology asset. First, it is a first-in-class molecule with high selectivity and pH independence. It is able to block VISTA function, which inhibits T-cell activities. PMC-309 is currently in the highly favorable competitive landscape. We signed a CDMO agreement with Thermo Fisher, a global bio company that will provide IND filing services. Then we plan to initiate a global phase 1 clinical trial of PMC-309, most likely in 2022. I'd like to explain the mode of action of 309. PMC-309 is an anti-VISTA antibody that restores immunoactivity of T cells by inhibiting VISTA interaction between the T cell and the MDSCs. Under normal body conditions, MDSCs act as immunosuppressants to prevent autoimmune diseases that results in hyperimmunity. However, MDSC also blocks anti-tumor effect of the T cells. We believe that PMC-309 provides a solution to this issue. PMC-309 can block the VISTA pathway between the MDSC and T cell, thereby fully activating the T cell anti-tumor effects. PMC-309 has a very outstanding looking competitive landscape. There are only 2 companies around the world. One is Curis and Pierre Fabre, and one is Pierre Fabre Pharmaceuticals in the clinical stage with their own VISTA molecule. The reason why there are so few developers in the market is because the very first VISTA molecule showed cytokine release syndrome in the clinical setting, which led other developers to stop or delay the development of VISTA molecules. This resulted in a very favorable environment for our PMC-309. It has very few developers in the market, and it is not too far behind in the development. According to the preclinical results of PMC-309, we were able to discover these strengths. PMC-309 shows synergistic effect when used in pembrolizumab in a tumor model. We were able to find that the combination of PMC-309 and PD-L1 antibody does show a synergistic effect, which means we can use these combos to treat patients who do not respond to the existing drugs. Also, PMC-309 has high specificity for VISTA. What I mean by high specificity is that the CRS, or cytokine release syndrome, happens when the molecule attaches to another receptor, resulting in hyperimmunity. However, it was found that our PMC-309 only has high specificity for VISTA and is expected to have safer rivals. One last strength of PMC-309 is that it remains active even in the low pH environment. In the tumor microenvironment, tumors release excessive VEGF signals to expedite the formation of blood vessels, which is used as a pathway to get nutrients for the tumor. However, blood vessels are formed in a hurry. They are disorganized and leaky, which results in inefficient delivery of oxygen and anti-therapeutic drugs. We were able to discover that PMC-309 still activates in the low pH environment. Now, we believe that this characteristic of PMC-309 will play a key role in immunotherapy. Now I'd like to move on to our next rising star molecule, PMC-403. PMC-403 is a first-in-class molecule, as it is the only antibody that directly targets type 2. It can normalize pathological blood vessels resulted from diseases and injuries. It can cover broad indications in oncology, non-oncology, and is a very versatile asset. PMC-403 was able to get its potential recognized by our partners. As we mentioned earlier, we signed an MCRADA with the NIH in the United States. PMC-403 is expected to enter global phase 1 clinical trial, most likely in 2022. Here is the mode of action of PMC-403. PMC-403 attaches to a Tie2 receptor, which is an angiopoietin receptor present on the endothelial cell. PMC-403 attaches to the Tie2 receptor, which results in the stabilization of blood vessels. The competitive landscape looks favorable for PMC-403 as well. Currently, PMC-403 is the only drug that directly targets Tie2 antibody and also a fully human antibody. Other competitors in the market are either peptides or the indirect Tie2 activating molecules. PMC-403 shows huge potential for eye diseases, such as diabetic retinopathy and age-related macular degeneration. Both diseases are caused by leaky blood vessels formed in the retina, which causes increased pressure within the eye. Currently, this market is mostly occupied by Lucentis, Eylea, Avastin, and Bellevue. However, we believe that PMC-403 can perform excellent in this setting because these drugs currently used in the market usually end up with the drug tolerance in the patients. However, PMC-403, with the mode of action that does not overlap with the existing drugs, will play an important role in this market. Here's our preclinical result of PMC-403. On the image on the left, we are able to see that PMC-403 reduces the artificial leakages. The result was quite comparable to that of aflibercept. This result can be seen in much more detail on the image with the image on the right. We can see that PMC-403 showed lower mean value in the overall size of leakage compared to aflibercept. One thing I would like to emphasize here is that PMC-403 is still under development and there is room for further improvement in efficacy. The development strategy of PMC-403 is just massive. We can expand its indications in eye, brain, respiration, immunology, and etc. We believe that PMC-403 will be our all-rounder drug, our most valuable asset. Now I will move on to our last preclinical pipeline, the PMC-402. There are 5 things to know about PMC-402. Just like PMC-403, it is a first-in-class molecule which binds directly to Tie2 receptor. PMC-402 has vessel normalization effect and immune cell infiltration. It shares similar characteristics with PMC-403, but it has better T cell infiltration. We believe this molecule has higher potential in oncology. We recently formed a CDMO contract with Samsung Biologics to get CDO services needed for IND filing. PMC-402 is likely to enter a phase 1 clinical trial in mid-2021. Now, I would like to explain the mode of action of PMC-402. The PMC-402 and PMC-403 sharing the common characteristics, their vessel normalization can play a key part in the oncology area. In the tumor microenvironment, the vessels are disorganized and very leaky, which results in inefficient delivery of anti-tumor drugs, oxygen, and nutrients, immune cells. Then it was our belief that by normalizing these blood vessels, we should be able to show better efficacy data in terms of tumor. Here is the preclinical result of PMC403, we cannot disclose the data of PMC402 as of now, but the data suggests normalizes brain tumor blood vessels in the mouse model. We can see that the leakiness in the preclinical model has been reduced. Also, we were able to discover that the combination of Tie2 activators and a PD-1 antibodies does show a synergistic effect, showing us that in the future, we can have clinical trials involving combo therapies. Also, we were able to discover that Tie2 activators show improved T-cell infiltration rate compared to other drugs. Lastly, I would like to talk about our future key milestones. First, we will initiate the phase 2 mTNBC combo study for olanzapine and pembrolizumab. The study will be ongoing in both Australia and South Korea. We will also be presenting the preclinical data of PMC309 at AACR 2021. Our e-poster will be available on April 10th. Then we will present the preclinical data of PMC403 at ARVO, the biggest eye and vision research association in the world. We will present these data in May. Lastly, we will initiate IND-enabling studies for all of our preclinical assets, such as PMC309, PMC403, and PMC402 within this year, and initiate the global trial within the next year. I'd like to thank you all for your time and interest in PharmAbcine. Have a wonderful day.